reason I came here is that I've always wanted to do research that we could potentially translate into human benefit, not just basic research. And the beautiful thing about this institute is we make incredible basic discoveries, but then actually try and translate it into something useful, and that's really why I'm here. So I wanted to sort of give you some of the science behind this stroke drug we're trying to develop, how it came about, why we're actually using venomous animals. There is a reason. And then tell you about some of the more recent developments and what we're using some of your donations to fund. So do I have a clicker here? I think I do. Yes. So Brandon has touched on some of this already. So stroke, I think we've all been touched by somebody in our extended family or circle of friends who've had a stroke, have been impacted by a stroke. As Brandon said, it is the second largest cause of death worldwide. It's the third largest cause of death in Australia. It's actually the leading cause of serious long-term disability, and the numbers are that 1 year after a stroke, 50% of survivors still require daily care of some kind to go about their normal activities. And the reason it's such a problem is that we don't actually have very many therapeutic options to prevent the brain damage that happens after a stroke. So there are two types of stroke, as you know. There's a hemorrhagic stroke, and that's when there's a bleed into the brain. And for that type of stroke, there's actually no therapeutic options whatsoever. So there are no drugs available for that type of stroke. For an ischemic stroke, there is one drug. It's called tissue plasminogen activator, or TPA. It's actually a clot-busting molecule. Because it's a clot-busting molecule, you actually can't use it for hemorrhagic stroke. It would actually worsen the situation. But this clot-busting molecule still has problems. It can still cause a bleed in the brain of a person with an ischemic stroke if it's used incorrectly. And therefore neurologists are actually very scared to use it. So less than 5% of stroke patients actually get that one available drug. So the vast majority of stroke patients get no medical treatment in the form of drugs at all. It's a singular failure of modern medicine, if you like, that we've not been able to develop anything to stop this brain damage. So I wanted to talk about what happens in a stroke. So 85% of strokes are ischemic, and that means there's a clot in an artery, a cerebral artery leading to the brain. And that part of the brain that's most affected by that lack of blood supply is called the ischemic core, and that's thought to, that part of the brain is thought to die really quickly, and the dogma that's taught to medical students is that it's unrecoverable. You can never stop that part of the brain dying after the stroke. It is going to die, nothing you can do about it. But then there's this spreading wave of destruction that happens quite slowly over hours to days to create this thing called the ischemic penumbra or peri-infarct zone. And because that develops slowly, it's been thought that if you had a drug that was suitable, you might be able to prevent that spreading damage and therefore minimise the brain damage. And that's what we set out to do, is to try and minimise the size of this peri-infarct zone. So what happens when you have this decreased blood supply to the brain is obviously a lack of oxygen, right? The brain, that part of the brain's getting no oxygen anymore. The brain has to change the way it actually harnesses its fuel, and the fuel of the brain is glucose. Glucose is the biggest user of glucose in our body. That's neurons, nerve cells are really energy hungry. But it uses oxygen to burn that fuel, and it can't anymore. It's got no oxygen. It has to burn the fuel in a different way, and it reverts to this ancient metabolic pathway called glycolysis, which is used by virtually all organisms, including bacteria. And that's fine, except the problem is the end product of that pathway is lactic acid. It's the same thing that happens when you're exercising really hard. You might recall when you're exercising really hard, you get that pain in your muscle. That's because you're getting a lack of oxygen there. Your muscle's also reverting to this glycolysis, producing lactic acid, and that's causing pain. But your muscle can clear that eventually. But your brain, now remember, it's lacking a blood supply in that part of the brain. It can't clear the lactic acid. And so your brain pH goes down. Your brain actually becomes acidic after the stroke. Amazing. The pH of your brain actually goes down a lot. And it turns out that on the end of your neurons, your nerve cells, are these little ion channels called acid-sensing ion channel 1A, a really horrible name. We just call it ASIC 1A, just like the shoes, ASIC 1A. And it turns out, as the name would suggest, it recognises that drop in pH. It recognises that the brain has become acidic, and it switches on. And when it switches on, unfortunately, it sends a message to that nerve cell to die. And that appears to be why a lot of brain cells die after stroke. It's because the acid activates that channel. The channel sends a signal to the neurons to die. And so we had a very simple idea, and that was, what if we could find something that inhibited that ion channel? I should say ion channels are just little things that sit on the membranes of your nerve cells to let ions in and out. Things like sodium, calcium, potassium. This one lets sodium in, as it turns out. What if we could find something that inhibited that ion channel, prevented it being activated in the presence of acid? Would we prevent the downstream brain injury? So that was the idea. But we needed a potent inhibitor, right? And so what did we do? We interrogated animal venoms. And that may sound like a crazy idea. It turns out, and I'll tell you why, we actually have the biggest collection of venoms in the world here in this building. We have venom from over 700 species of venomous animals. And you might say, why is that? Well, you notice they're all actually invertebrates. There's no snakes, there's no lizards, there's no platypus. And the reason for that is we're interested in drugs that target the nervous system. And all of these invertebrates target the nervous system of their prey. Snakes tend to be hemotoxic. They tend to target the cardiovascular system. Probably not good for a stroke or heart attack, right? Not the thing you want. So we work with these sort of guys. And so when we were looking for an inhibitor, we can interrogate all these 700 species. So the winning molecule could have come from any venomous animal anywhere in the world. And the really remarkable thing is the winning molecule came from this guy, the Fraser Island Funnelweb Spider, just a couple of hours north of here. So we work with this spider a lot because Funnelweb spiders, you might know, like to dig holes in the ground. They live at the bottom of their burrow, and if they're living in a clayey soil, they're quite tough to dig up. But because Fraser Island is the biggest sand island in the world, it's much easier to dig them up. So that's me and a former postdoc from my lab, who's now at James Cook University, digging for some funnelwebs on Fraser Island. And that's what it looks like. It's quite a nice spider. And this is the molecule we found. So the molecule we found is a small protein, or peptide. It's to give you an idea, it's about 1.5 times the size of human insulin. It's about the same size as a human hormone. But here's the incredible thing. When we started the project, we knew there was one small molecule drug that inhibited this channel. It's called amelioride. It's an antidiuretic drug. It's commonly used as an inhibitor of this channel. But this molecule, which we call HI1A, inhibits the channel with 20,000-fold higher potency than that small molecule. So it's a brilliant inhibitor of that channel, incredibly potent. So it was exactly what we were looking for to test our hypothesis. The question was, this blocks the channel, does it block the brain damage? And so this is the experiment we did. We took rats who'd had a stroke and we gave them the drug initially 2 hours after stroke. And that's really important, that you show that the drug can work well after the stroke starts, because more than 60% of people take more than 2 hours to get to the hospital. So you want something that will work many hours after the stroke. So we started with 2 hours and we looked just at the brain damage in that peri-infarct zone. Remember, that was the part that's supposed to be recoverable. And you can see that by comparing the red and the green, we got an 83% reduction in the amount of brain damage. It was quite phenomenal. So then we went to 4 hours. What happens if we gave the drug 4 hours after the stroke started? That's the maximum time at which you can give that currently available drug, TPA. And you can see we're still getting a almost 90% reduction in the amount of brain damage. So then we stepped off the cliff, as Brandon would say. We went to 8 hours. There's nothing available currently that works beyond 4. We went out to 8 hours and said, what happens if we go way out to 8 hours in a rat, which would be even longer in a human? Turns out we still got a 65% reduction in the amount of brain damage. So still very significant, even 8 hours after the stroke. So that's the peri-infarct zone, the zone that was thought to be potentially recoverable, and it clearly is. What about this ischemic core? Which, remember, the med students are taught not recoverable. Well, in fact, we see a reduction in the ischemic core damage at all time points. That was completely unexpected. So we don't think the ischemic core is unrecoverable. We think with the right drug, the right timing, you can actually prevent brain damage in that ischemic core region. That was really exciting. But the other thing we wanted to look at was, well, what's the quality of life for the survivors? It's one thing to survive the stroke, but it's another thing to have good quality of life after you have the stroke. So we looked at two things. We looked at what we call the neurological performance of the rodents. their ability to do certain tasks, and we looked at their coordination, that is how well their muscle activity is coordinated. So this neurological score goes from 0 to 6. It's a composite of lots of tasks. 0 means a perfectly functioning rodent. 6 is effectively a paralysed rodent and can't do anything. So you can see 72 hours after stroke, these animals are very highly impaired. And this is what happens to the animals that had the stroke, but got the drug 2 hours later. We've almost returned their neurological function to normal. So not only have they survived, they've survived with very good neurological function. The other thing we tested was their motor performance, and to do that, we get the rats to walk down a ledge beam, and rodents are really good at climbing activities. So you can see before the stroke, they never make a mistake. They always manage to walk along the beam into their nest, no problems. So you see 72 hours after the stroke, they're falling off, about 80% of the time or more. And if they get the drug though, you can see their motor function's restored almost to normal. So they're able to walk down this beam. So why is this really important? Why do we think this is an important development? The first thing is, this is not a clot-busting drug like TPA, which means it should be perfectly fine for people that have a hemorrhagic stroke. Even if it doesn't help them, it certainly won't hurt them. Why is that important? That's important because at the moment, you can't treat a stroke patient until you get them to hospital, image their brain and find out whether it's hemorrhagic or ischemic, because if it's a hemorrhagic, you can't give them anything. If it's ischemic, you might be able to give them TPA. But it means you've got to get them to hospital, and that could take a long time. But because this doesn't induce bleeding, you can give it to the patient straight away. It means that you should be able to, first responders should be able to deliver the drug straight away. Why is that important? Because after the stroke, you lose 2 million nerve cells per minute. So the sooner you can get some protection of the brain, the better. So it'd be really incredibly helpful if first responders could give the drug to the patient before you got them to hospital. The second thing is, as I've shown you, there's a long treatment window. We went out to 8 hours, which would translate to more than 10 hours in a human. That's particularly important in somewhere like Queensland. If you're way out in regional Queensland or you're in the Torres Strait and you need to get to a East Coast hospital, that can take 6 or 7 or 8 hours. And we've shown that even if you give the drug at 8 hours, you can still protect some of the brain. So even those people that couldn't get the drug straight away and took a long time to get to the hospital, they could still get some benefits. So there'd be better outcomes, we think, for people living in regional areas. The thing that you probably haven't heard about is that we recently, with Nathan Palpant here, and this is the beauty of what Brandon talked about, we have a diversity of people in this building with different interests and different expertise. And it turns out a few years ago we recruited a young fellow from the United States whose expertise is cardiac physiology. And I said to Nathan Palpant, what's known about this channel in the heart? And he said, well, nothing. I said, well, is it important then in the injuries that the heart gets, say in a heart attack or a cardiac arrest? He said, we don't know. So we looked, and it turns out it's equally important for the heart. And so what we've shown is the drug can protect the heart during a heart attack. And what we've been particularly interested in, working with John Fraser here at the Prince Charles, who is in charge of the heart transplant program here in Brisbane, and Peter McDonald down in Sydney, is we can actually protect donor hearts that are going to be used for transplant. It turns out, and I was shocked when I heard these statistics, it turns out that only 1 in every 4 donor hearts makes it into a patient. We actually throw away 75% of human hearts. And the reason for that is that, and I was shocked by this as well, once the heart's taken out of the donor, the standard of care is it's put in a Ziploc and put into an esky. That's it, the standard of care for a human heart. It becomes ischemic straight away. It's got no oxygen, it's got no blood supply. It suffers the same damage the brain does during a stroke, except now instead of nerve cells dying, it's cardiac muscle that's dying. And we've shown that the molecule can protect that from happening as well. So we think it has applications in potentially heart attack, out of hospital cardiac arrest and heart transplant as well. So we think the drug addresses this desperate need we have for a drug that can protect the brain after stroke. We think it has additional applications in heart attack, cardiac arrest and heart transplant as well. And these are what we're using the funds that you've kindly donated at the moment. The drug is very complicated chemically, it's difficult to make, and so we're trying to develop a better manufacturing process. The process we use in the lab at the moment has been great for small animal experiments, rodents, but not so much for large animals like sheep and pigs, which are much larger. So we're trying to improve that, and we're also trying to work out what is going to be the optimal dose for stroke. The initial experiments we did was just a one single dose, and it could be that multiple doses, continuous infusion, high dose, could give you even better results. We don't know. So we may be nowhere near the optimal dosing for the drug yet, which is exciting. So that's what we're doing at the moment. So I'll leave it there, and I'm very happy to answer any questions you have. Once again, thank you very much for your support. I really appreciate it.
So stay there.
Yeah.
Please ask Glenn.
Yeah, please.
Questions?
Questions?
He doesn't bite.
Yes.
And,
My mother had a stroke in a nursing home with her physiotherapy there. If there was something like an EpiPen.
Brilliant, great question. Yeah, that's it.
You know, can you do that?
Yeah, so that's it. That's the holy grail for us. Is we'd love to develop this just like an EpiPen. So it could be there in nursing homes, it could be there in ambulances, aged care homes. So somebody's having a stroke, boom, EpiPen with the drug, they get immediate protection, or a heart attack, they get immediate protection. So that's our ultimate goal. It's a great question. Thank you. Very good.
Historically, what sort of timeframes is it to get to market?
Yeah, it's a good question. It's hard to predict. It's certainly we're going to be measuring in years, not months. We think in this case there's an easy route to get it into clinical practice for physicians to get comfortable with it, and that is heart transplant. Heart transplant, an easy one because you'd actually have the drug with the donor heart, it actually get washed out even before it gets put in the patient. Would give the clinician some confidence that this is a good drug, that it works, and then moving to stroke after that. So we're thinking three years is probably the time frame, 3 to 4 years to get it through clinical trials. So that's a long, slow process, unfortunately. But it's yeah. Yep, please.
Do you see that you could use it in, you were saying, stroke affects children and all the way through?
Yes.
Would you be able to use it for all?
Yes. So we've actually just started a collaboration with a group down in Melbourne who works on postnatal stroke. So it turns out a lot of very young children have stroke, and that's incredibly unfortunate. The only good thing from our point of view is the fontanelle hasn't closed, so delivery of the drug to the brain is actually very easy in those children, and so we're trying to do a project where we can deliver the drug directly into the brain of those kids and see if that has an impact. So yes, it should be able to be delivered to any age group. Yeah, it's a great question. Thank you. Yes?
What made you ever thought about, think that venom of the spiders would be useful?
Yeah, look, we work with venoms generally because, technically we're interested in nervous system disorders where the underlying problem is an ion channel, these ion channels that I talked to you about. Turns out these ion channels are now the third biggest target of human drugs. So a huge number of human drugs target these, and we're learning more about them all the time. It turns out the best source of things that target those ion channels is venoms, because that's what these venoms do. They target ion channels in their prey and predators to either cause pain or to kill the prey. For example, spider tries to kill an insect by incapacitating its nervous system, basically by hitting those ion channels. So it's a really good source of ion channel drugs, and that's why we use them. It was actually quite serendipitous in this case. We were working on a different spider venom drug that was discovered by somebody else, and we had a girl in the lab, a really smart PhD student, had a completely different project. She wasn't working on stroke. She was just working on that Australian Funnelweb Spider, and she was trying to understand why it was so complex. It turns out the venom of the Australian Funnelweb Spider is about the most complex venom of any animal in the world. We think it's it's got more than 3,000 molecules in it. And we don't know why. It's incredibly complex. And she was trying to understand how did it evolve such an incredible complexity. And in doing that, she came to me and said, "Oh, look, I found a molecule that looks like your stroke drug." And she showed me, and I said, "Wow, that is interesting." And it was similar, but twice as big. It looked like a duplication of the one we were working with. And I said, "Well, we should try it." And it turned out to be much better than the one we were working with. So it was a great sort of story to other students to say, be aware of what's happening around you. You never know when your project will intersect with somebody else's project, which it did in this case. So we were just very lucky that she found that, because she could have just skipped over it. So I was very lucky. Yep.
Is there any difference in the Funnelwebs from Sydney or elsewhere in the country to Fraser?
Yeah, look, I think they mostly have the same sort of repertoire. The lethal toxin can vary a little bit. We know exactly what that lethal toxin is. So of those 3,000 molecules in the venom, there's only one that kills us. And we know exactly what that molecule is. We know exactly how that molecule works. It varies a little bit between each of the, I think there are now 35 species of funnelweb spiders. And we're finding more all the time. We've actually got downstairs a new species that's just been found in Tasmania. Amazing. So we still haven't found all the species of funnelweb spiders, but they all, I think, are going to be dangerous to humans to, a greater or lesser extent because of that one molecule. And I should point out too that the reason we like working with invertebrate venoms is for the most part, they're completely harmless to humans. We're just unlucky in Australia that we happen to have a lot of deadly venomous animals. We don't have any more venomous animals than Brazil, for example. Brazil has just as many venomous animals as Australia, it's just they don't have very many that can kill humans, and we seem to have a lot of those. But for the most part, there's more than 100,000 species of spiders, only 4 of them can kill you. Just unfortunate that 2 of them live here in Australia, which is the redback and the funnelweb. But even the very biggest tarantula, the Goliath tarantula, is that big. It's as big as a dinner plate, completely harmless to humans. So most spiders are completely harmless. And that's why we like working with them. We know that there's not intrinsically anything in the venoms that will harm us. So they're actually good fun. You'll actually get to meet some of these spiders later, I hope. Yeah. I don't anymore. Yeah, no, I get the students to do that. We've only lost a few. That's right, yeah. No, it's actually the funnelwebs are the easiest spiders to work with of any spider we work with, it turns out, because one thing is they don't climb. So we work a lot with tarantulas. Tarantulas have little pincers on their legs, their feet, and they can actually climb up surfaces. And actually, I was talking to this with a reporter a few weeks ago, and I had a tarantula in a container, and I had a long pair of forceps, which we usually use when we're doing media stuff. And I turned around and I said, look, the funnelwebs are easier to work with because they don't climb, but the tarantulas can. At that very moment, it ran up the forceps and jumped out onto the bench, and she went, ah! It was just perfect timing. It was brilliant. But the funnelwebs, they get so excited that they might be able to kill you that they actually start to drip the venom off their fangs, and you can just suck it off with a pipette. You don't even need to do anything. All the other spiders, we actually electrically stimulate their fangs to force them to squirt it out. But funnelwebs, you don't even have to do that. It just drips off their fangs.
So have we been able to artificially produce this venom?
Yeah. Not the whole venom, but the molecules we want. Yeah, great question, yeah. Because they're just little peptides, they are encoded by a gene. And so we can just take that gene and in our case, we put it into a bacterium, just common old E. coli, Escherichia coli, and we direct the coli to make it. So that's how we make it in the lab. We just don't get very much that way. We're trying to move it into a yeast, so you can grow it in big fermenters like beer. That's usually a better way to scale up to make lots. But the other way is chemical manufacture, and we're trying that as well. So that's what some of this money is being used for. What is the best route of manufacture? And the other thing we're trying here, which is we really like, if it worked in the long term, because we'd like this to be available all around the world at a cheap price, is Ben Hankemer, and again it gets back to this incredible diversity of people here. Ben Hankemer here runs the lab next to mine. He's interested in solar biofuels, but using algae. And so algae, the thing about algae is it's completely scalable. You can grow them in massive ponds. So we get, and you can grow them in your swimming pool, that's right, yeah. Cheap, easy, just use the sunlight. We're trying to instruct the algae to make this drug. And that'd be really cool, because then you could make it anywhere in the world and you can make it really cheaply, be really super scalable. So that's something that we've just found out today. We got a grant from the National Health and Medical Research Council to try and do that. So yes, a rare success in the current environment.
Funnelwebs all over the world? Or
No, only in Australia. We're the only lucky country to have funnelwebs. Yeah, no, they're only here. There's some closely related ones up in Papua New Guinea and Southeast Asia, but no, funnelwebs only here.